This program was produced by the Chemical Heritage Foundation. Our mission is to collect, preserve, and make known the history of chemistry, chemical engineering, and related sciences and technologies.
I think the important thing to realize is that the things that seem so exciting today, those discoveries are already happening. Really, you want to put yourself in the position where you'll make the discoveries for tomorrow. I think that's really the important thing, is to learn fundamentals, is to do cutting-edge science and learn how to do it and learn how to ask big questions. I'm Bob Langer. I'm a professor at MIT. I'm a bioengineer. I'm probably good at generating ideas. I think I'm a pretty good teacher. Those are a couple things.
This is the story of a teacher, engineer, inventor, entrepreneur, and pioneer who carved an untraditional path in the field of chemistry, following his heart and mind with the simplest of goals: to do good with science. Robert Langer is widely regarded for his contributions to medical and biotechnology. It is estimated that his work has impacted 2 billion lives. He holds over 1,000 patents worldwide, which have been licensed or sub-licensed to over 250 pharmaceutical, chemical, biotechnology, and medical device companies. He's launched 24 startups, making an eclectic variety of things from tumor-finding nanoparticles to synthetic spinal cords, all originating from the same lab. He is recipient of countless awards, including the National Medal of Technology and Innovation and the National Medal of Science. And although his path to the laboratory at MIT wasn't always a clear one, his focus on helping others was unwavering.
My parents weren't involved in science. My dad, actually, he ran a liquor store. He was a small businessman. He ran a liquor store in Albany, New York. When I was a little boy, both my dad and my grandfather, they'd play a lot of math games with me, and I think that got me interested in math. And then they also gave me these Gilbert sets, this Gilbert Erector set, Gilbert Microscope set, and Gilbert Chemistry set, and I love those. I think the Gilbert Chemistry set I really enjoyed. We even set up a little lab in the basement, which, you could have reactions occurring where, you'd have a couple different solutions and you'd mix them together and they change color, or you could make rubber. I think it was those sets that helped get me interested in chemistry in particular. My mom was a homemaker and basically took care of me and my sister. She's just a lovely, nice, sweet person. She worries a lot, but she's just a very, very nice human being who cared a lot about people. Hopefully a little bit of that rubbed off on me. I like to think I was a pretty regular kid. I mostly would play baseball, football, and basketball in the neighborhood. I don't think I was a very best behaved kid in school either. Looking back at it, I'm sure I had attention deficit disorder. I had tremendous difficulty paying attention in class. I think I was probably pretty regular. I probably wasn't that great a student either.
At the Milne High School in Albany, Robert excelled in math and science, and while his career goals were uncertain, his family encouraged him to study engineering in college. As a freshman at Cornell University, Robert enjoyed his chemistry class the most and decided to major in chemical engineering. Another nice surprise at Cornell was a job as a teaching assistant, which revealed a natural affinity for teaching.
Yeah, I loved being a teaching assistant when I was at Cornell. I would spend a lot of time working with the students, and I think they really appreciated it. I'd never done much teaching before that. It was very exciting to me, and it was something that I could see I was good at. When I was at Cornell, I didn't have that clear an idea of what chemical engineers did. And when I applied to different graduate schools, I was fortunate that I got into a number of them. And the thing I liked the best about MIT's was that since I didn't know what I wanted to do, it seemed like the most diverse.
Although Robert was a graduate student at one of the nation's premier institutions, he wasn't blind to the significant problems facing the surrounding neighborhood.
Cambridge in the 70s was an interesting place. On the one hand, it had Harvard and MIT, and on the other hand, it had the highest high school dropout rate of any city its size in the country.
Cambridge offered Robert a chance to pursue his academic interests at MIT. It also offered Robert a transformational teaching experience at an alternative high school nearby called the Group School. The group school was established primarily to serve students from working class and low-income backgrounds. It operated under democratic school principles, an educational ideal in which democracy is both a goal and a method of instruction.
The school was aimed at trying to help those kids who had dropped out get back and learn things, and it was very successful. I mean, I think the people working there were very dedicated. My job was actually, I was in charge of the math department and the science department. That was very, important time in my life. Basically, I'd probably do two things. I worked in the lab and I worked at that school.
At the group school, Robert worked to develop a math and chemistry curriculum for high school students that was fun and academically solid. At MIT, he pursued his graduate work, doing different kinds of experiments aimed at ATP regeneration. Adenosine triphosphate, or ATP, is an energy-bearing molecule found in all living cells. His dissertation, titled Enzymatic Regeneration of ATP, made him a player in the then new fields of biochemical and biomedical engineering. Robert earned his Doctor of Science degree in chemical engineering from MIT in 1974. That year, he found himself at a career crossroads.
I really didn't know what I wanted to do. All my classmates, pretty much would get these high-paying offers from oil companies, and they, at that time, they would interview at MIT. Other chemical companies did too. And there was a lot of job offers, and so I thought that's probably what I'd do too. I just thought I'd go along and do what everybody else did. But when I went on the interviews, I wasn't very excited about it. It's funny, when I was at the companies, the people were nice, and I guess I could sort of see myself maybe doing it. But then I'd come back home and I'd say to myself, I really want to do something that, at least to me, would have more impact.
Robert received 20 offers from oil companies, including Shell and Chevron, and 4 from Exxon alone. But he couldn't interest himself in fuel technology. The experience at the group school had affected him deeply. He was searching for a career where he could help others more directly.
Then one day, when I was walking into the lab, one of the postdocs, Barry Bunow, said to me, he said, "Bob," he said, "there's a surgeon in Boston named Judah Folkman." And he said, "Sometimes he hires unusual people."
Judah Folkman was a path-breaking cancer researcher, a professor at Harvard, and director of the vascular biology program at Boston Children's Hospital. Robert wrote to Dr. Folkman expressing interest in his work, and Dr. Folkman offered him a job in his lab.
He had a vision that if you could stop blood vessels, that might be a whole new way of stopping cancer from growing. And that just seemed so exciting to me, that if you could do that, that really would make an impact, and a big impact, and I was very excited about that. So it was the lowest paying job by far, but it was the one that I felt was, where I could someday maybe make the greatest impact, so I took that one.
Robert was the only chemical engineer in the hospital, because at the time it was rare for an engineer to be working in a surgical lab. However, Dr. Folkman was pursuing an unorthodox approach to cancer research and was willing to employ postdoctoral fellows without conventional medical or biological training.
Dr. Folkman was a visionary person. He was a dreamer. He thought anything was possible. He never gave up. I mean, he was a fantastic role model. He worked all the time. I remember some nights leaving the hospital at 2 a.m., and he would be coming in. He was just a remarkable person to work with. Being a postdoc, that changed my life. I mean, that caused a fundamental shift. When I was there, for me, in a way, it was almost being like a kid in an intellectual candy shop, because being the only engineer there, I would see all these surgery problems, and they were trying to solve it certain ways, but I thought, gee, you could solve this by chemical ways or engineering ways. And so I got all kinds of ideas, new ways of drug delivery, new ways of using enzymes to do things in the body, new ways of growing tissues and organs, new types of materials.
Dr. Folkman believed that the spread of cancer and the growth of tumors could be controlled if angiogenesis, the process by which new blood vessels are created, was arrested.
The big goal was, could you understand that, and could you stop it? Because if you could stop it, that might lead to a whole new way of treating cancer.
He charged Robert with the task of finding substances to inhibit the creation of new blood vessels.
We did thousands of experiments, and it took a lot of persistence. Most of the time, of course, I'd fail.
Robert worked with Dr. Folkman to isolate the first angiogenesis inhibitor, a macromolecule. Macromolecules tend to be broken down by digestion and blocked by body tissues if they're injected or inhaled, so finding a delivery system for them is difficult. Robert's idea was to encapsulate the angiogenesis inhibitor in a non-inflammatory synthetic polymer microsphere that could be implanted in the tumor and control the release of the inhibitor. He eventually invented a polymer system that worked. His discovery laid the foundation for much of today's controlled-release drug delivery technology.
Right now, when you take a drug, the drug levels sort of peak and trough. They go up, they go down. Peaks can cause toxicity. The troughs, the drug doesn't do anything. What controlled release is basically taking materials, which could often be a polymer, and you'd put a drug in it, and it could deliver the drug at a steady rate for a long period of time. That could be anywhere from a day to years.
Although this technology now spans many fields and includes pharmaceutical, food and agricultural applications, pesticides, cosmetics, and household products, Robert's research on controlled release was not well received when he first published his findings. Scientists felt the molecules were too large to be transported through solid polymers.
There was an enormous amount of skepticism. People ridiculed it. That went on for quite a few years. It was actually very damaging to me as I tried to get job offers, as I tried to get promoted, and as I tried to get grants. But that sometimes happens in science. Over time, lots of labs repeated it, even though it didn't make sense to polymer people or chemistry people. There were a lot of biology people that wanted to use it. Hundreds and hundreds of labs would use it around the world, and they would publish on it, and it worked for them too. And then, of course, eventually the pharmaceutical industry would use it. So I think all those things, what we did, what others did, that would whittle away at this, and now it's, I think, widely accepted that this does work.
He also worked with Henry Brem of the Johns Hopkins University Medical School on a drug delivery system for the treatment of cancer to deliver chemotherapy directly to a tumor site. The wafers or chips that he and his teams have designed have become increasingly more sophisticated and can now deliver multiple drugs to a tumor site. Robert is regarded as one of the founders of tissue engineering in regenerative medicine. A colleague from Dr. Folkman's lab, Jay Vacanti, interested Robert in combining three-dimensional synthetic polymer scaffolds with living cells to create new tissues and organs in the laboratory.
Well, I first met Jay Vacanti when I was a postdoc in Dr. Folkman's lab, and he was a surgeon there. About 3 years after that, he became head of the liver transplant program at Boston Children's Hospital. And I remember him coming to see me and telling me about the little children he was trying to treat who were dying of liver failure. And that really the only way he could treat them was that somebody else would die, and they'd take the liver from that person and do a transplant. And so he asked me, could I help him? Could we make an organ from scratch?
The idea of creating something that could save lives and not have to depend on an organ donor appealed to Robert. The challenges were plenty, and he set to work. Robert quickly abandoned his initial idea about making a two-dimensional polymer wafer on which cells could sit.
One day I remember Jay was at Cape Cod, and he saw seaweed, and he was looking at that sort of three-dimensionality, and I remember him calling me up and saying, could we make polymer structures like that? And I said, sure we can, and we did. And that would lead to our early patents and papers in that field.
Robert and researchers in the Langer lab have made advances in tissue engineering, such as the creation of engineered blood vessels and muscle tissue. Bioengineered synthetic polymers provide a scaffolding on which new skin, muscle, bone, and entire organs can be grown. With such a substrate in place, victims of serious accidents or birth defects could more easily grow missing tissue.
There are no treatments for many of these diseases. It's not like a drug could create a new tissue or organ. So this has been very exciting to see the potential of the field of tissue engineering.
At MIT, the Langer Lab is a place where innovation and creative thinking thrive. There are over 100 students working in Langer's lab today. They're working on all kinds of nanoparticles that could target different parts of the body, and nanoparticles that could be used for more rapid diagnostics. They're also working on new kinds of implantable microchips that can be regulated by remote control. Not unlike his experience in Judah Folkman's lab, the Langer Lab pulls together researchers from a variety of disciplines, including biologists, chemists, physicists, electrical engineers, and practicing medical doctors from all kinds of fields. This incredibly diverse group of researchers gives the Langer Lab the flexibility to dream big and take ideas all the way from inception to clinical trials.
Basically, want it to be kind of almost this idea factory where I might generate ideas, and I try to encourage the postdocs and graduate students to generate ideas, and they do. And we try to pick the best ones and go with them and try to make the biggest impact we possibly can. What I try to think about are things that have the potential to change the world.
As he found increasing success in his research, Robert also became heavily involved in entrepreneurial ventures. Many people approached him about starting companies. He encourages his students to adopt his entrepreneurial attitude as well, giving them plenty of guidance on patenting inventions and forming startups. Robert says he looks at his students as an extended family. Many of his former students now lead companies or laboratories of their own and advise governments on medical technology. As a colleague has said of Robert's students, they come away thinking that nothing is impossible. Polaris Venture Partners, a fund that has supported many of the companies that have had their origins in the Langer Lab, estimate that research done by Robert and his colleagues ultimately affects 2 billion people.
I'm not the kind of person that that's just curious. I valued science ultimately for the good that it can do. And I've gotten a lot of satisfaction out of trying to create science and engineering that can do good, rather than just be very curious. And so I think it's that thread that goes through my own career.